Hey guys, welcome back to another episode of Honors Biology Room 500 with Mr. Mann. Welcome back. Today we're going to talk about DNA within biology. So we're going to identify the substances of the gene or what makes up genetic material. So we should be able to answer these three questions. What experiments lead to the discovery of DNA as the genetic material, and what is the basic structure of DNA? Also, what is the basic structure of eukaryotic chromosomes, which we'll discuss very, very last. So, if we remember George Mendel from 7th grade, and looking at what he did in the 1900s with the pea plants, and looking at how traits are passed down to offspring within his fractions or percentages due to homozygous, heterozygous, we didn't have the know-how to solve for what the molecule of inheritance was. So that's what we're going to be talking about with these first three scientists today, or these first three experiments. And so the following experiments allow the scientists to determine that DNA was a molecule of inheritance. And we knew that genetic material was carried on chromosomes, and that chromosomes were consisted of both DNA and proteins. But we didn't know between proteins and DNA which ones passed genetic material to the offspring. So the first major experiment was performed in 1928 following the pneumonia outbreak. And as you can kind of see with especially this year, we can see that outbreaks cause a lot of scientific exploration in order to solve or remedy the outbreak. And so there were two types of pneumonia at the time, a virulent or disease-causing strain, or the smooth strain, or the non-virulent or the non-disease-causing strain, or the rough strain. And so Frederick Griffith was a British bacteriologist, and he questioned whether the ability to cause the disease was a direct result of whatever the product was making the bacterial colonies smooth, or whether the rough strains were less capable of establishing disease for just some other reason. And so his hypothesis was that bacteria had to be living to cause disease, and heat treating to kill the bacteria would prevent the disease because, again, the school of thought was, at that point, was proteins were more favorable to kill, to pass genetic material. And so if we killed, by using heat, the bacteria, we would denature the proteins in order to then prevent the disease. And so there are three types of, or four different variations within this. So we have the smooth strain, or the disease-causing. We inject it into the mouse dies. In the rough strain, or the non-disease-causing, we inject into the mouse lives. If we heat kill the smooth strain, the mouse lives. And then if we combine the rough strain and heat killed smooth strain, the mouse dies, which brought up to an interesting question of why would that happen if it didn't happen the first time, or if we heat killed smooth strain, why the mouse lived, but when we combined it with the rough strain, why killed the mouse? So Griffith knew that heating the bacteria denatured the proteins. Okay, thus he concluded the genetic material from the virulent strain survived, and what it did was it incorporated itself into the non-virulent strain, or changed the genetic makeup. But he concluded that proteins were not genetic material in 1928 due to the denaturing of proteins through the heat. So Oswald Avery, he was a Canadian-born American physician and a medical researcher, and he decided to take Griffith's experiment one step further. He knew that something within the cells was transferred from one bacteria to another, but he couldn't figure out what. So he hypothesized that he could find out what it was by systematically transferring the different possibilities into a non-virulent bacteria. So what he did was he transferred parts of the S strain cells, or the virulent bacteria, into certain sections and find out what the R strain cells tested. So When he transferred the RNA, R-strand cells were still there. When it was protein, we put in the protein into the R strain, it was still R strain. Same thing with lipid and carbs, but when he put in parts of DNA from the S strain, it mutated into S strain cells. So that the molecule, or the conclusion was the molecule that carries the inheritable information is DNA. Now, this was in 1944, and he concluded that DNA was genetic material. However, popular opinion at that time still favored proteins as hereditary material, which is very unfortunate because we had two scientific studies that backed up that it was DNA, or at least not protein. So then we had Alfred Hershey and Martha Chase. And they worked together to finally exclude protein as hereditary material. Now, Hershey and Chase attempted to perform the experiment in a different manner. What they did was they radioactively labeled proteins and DNA and tested two competing hypotheses. The first hypothesis was that protein was genetic material, and the transformed bacteria would contain radioactive material from proteins. And if DNA was genetic material, then the transformed bacteria would contain radioactive material from DNA. And so you do that by using two different radioactivities. We used one through phosphorus and one through sulfur. This phosphorus was with DNA, the sulfur was within proteins. So we use this bacteriophage, and I'm going to blow this up here, I think. I cannot. This bacteriophage here, let me use my pointer. Within the sulfur label, the protein capsule. So everything here in red is protein, except for this genetic material inside. While the phosphor label only labels the DNA inside the bacteriophage. So when they inject the genetic material, when they blend the genetic material, and then when they centrifuge the genetic material inside the cells, there was no dye on the cell, which means that there is no protein within genetic material. While there was genetic material found in these red strands here, said that there was genetic material inside the bacteria. So Hershey and Chase concluded that DNA, not protein, was the genetic material. They determined that protective protein coat was formed around the bacteriophage, but the internal DNA is what conferred the ability to produce offspring inside bacteria, and that was in 1952. I would also make sure that you know scientists and what came about from each scientist. So, we figured out what makes up DNA, but we didn't understand the structure of DNA. And so, Levine determined the basic structures of nucleotides in the 1920s, and he found out that it's made up of a phosphate group, a sugar, and a nitrogen-containing base. Now, one of the biggest differences between DNA and RNA is that DNA contains deoxyribose sugar, while RNA contains ribose sugar, or in this case, deoxyribose nucleic acid and ribose nucleic acid dealing with the sugar that makes up the compound here. Now, nitrogen-containing bases are the only difference between the nucleotides, as we see here. So we have our pyrimidines, or a single ring with thymine and cytosine, and we have our purines, our double ring between adenine and guanine. Now, I'm not expecting you to know the structural formula, but the biggest thing is it makes sense with single rings and double rings when we look here in a second. Now, there are two types of bases we've already seen, purines and pyrimidines. The other one we haven't seen yet is uracil. Now, uracil also replaces T within RNA. So when we talk about RNA and transcription and translation on Thursday/Friday, you'll know with uracil replaces thymine. So in 1949, Chargaff observed that the percentage of guanine was equal to the percentage of cytosine, and the percentage of adenine was equal to the percentage of thymine. This finding that there was a basis for the base pair rules, or the Chargaff base pairs. And the rule states that A always binds with T, and C binds with G within DNA. Because the pyrimidine to single-ring pairs with the purine, the helix has a uniform width due to shape. And we see our little A's and T's and G's and C's. So, in 1953, there was these two guys named Watson and Crick, and they were credited for determining the structure of DNA. They realized that DNA was the makeup of a double helix and made up of a sugar phosphate backbone on the outside with the bases on the inside. However, they did not know the shape of what DNA was. All they could figure out was the backbone, but couldn't figure out how it would intertwine together. This came with the help of Rosalind Franklin and Maurice Wilkins in 1952. Franklin had an X-ray diffraction experiment suggested that DNA was a double helix with a specific width. And so as we see this X-ray here of what she actually came up with, or what she took a picture of with her DNA. So this is a two-dimensional model. So we can see here, we have that wraparound when it comes to DNA. This is our picture that we usually see with our double helix, and then we have our base pairing inside the double helix. But a two-dimensional model, if we keep going within this capacity, to make it a three-dimensional model, that's what we would see. And so in 1962, Watson and Crick and Wilkins received the Nobel Prize of Medicine, as Franklin had died in 1958 and did not share the prize. Instead, Cambridge named their genetics institute after her. And it was due to the help of Rosalind Franklin and Wilkins in order to come up with this shape that it was imperative in order for us to figure out what the whole structure of DNA was. Now, DNA is structured like a spiral staircase. The sugar and phosphate molecules form the handrails or the backbone, and each turn of the DNA helix has 10 nucleotide base pairs. Now, the sugar phosphate backbone is connected by covalent bonds, and the nucleotide bases are connected by hydrogen bonds. And the biggest thing for hydrogen bonds is they're very easy to break with that DNA helicase that we'll talk about on Thursday when we talk about transcription and translation. Now, the orientation of strands of DNA is another unique feature. The top rail is a 5', and the bottom, or 5' on the left, top left, and on the bottom left is 3'. While it's anti-parallel, so it's 3' on the top right and 5' on the bottom left. And the biggest thing within this is due to the bond within the sugar, how it's numbered. So you see 0, 1, 2, 3, 4, 5. Okay, that fifth bond is where that 5', while here with this hydroxyl, it's the 3' part of the sugar. And then it's flipped on the other side, running in anti-parallel direction. Now, the base sequence of DNA is in the order of nitrogen bases on the chain of DNA. And each person has a unique set of base sequence, which the whole is called a genome, or what makes you you, which will make more sense on Thursday. The DNA of a eukaryotic cell ranges from 51 million to 245 million base pairs, which is about 5 centimeters long. The eukaryotic DNA is arranged in tight coils and around a group of proteins called histones, which we've talked about previously, to making chromosomes. And the DNA and histone complex is known as a nucleosome. I would know that word. And so then you should be able to describe the two characteristics of DNA needing to fulfill its role as genetic material. In our next section, we'll talk about replication of DNA. Okay? So that's it for me. I hope you guys have a great day. And if you have any questions, please let me know.